Okay, so what we have now is part 2 of nervous system 1. So we're still on chapter 10. Remember that you're going to have several more of the nervous system, 2 more chapters on the nervous system, but this is still part of chapter 10. And so we're going to look more into this whole thing with the nerves. Here's the dendrites, you got an axon coming across here. And so how is it that information gets from the dendrites through the cell, out an axon, and to another cell? All right. So the transmission then is something we call a synapse. And we talked about synapse before when we talked about muscle cells. Because remember, the muscle cells have to get their information. They get their information from a nerve cell. So when you look up here in this corner, here's the nerve cell bringing an electrical transmission. And then that information has to jump from that nerve cell to a muscle cell in the case of the last chapter, but it could be any cell. It could be nerve cell to gland, or nerve cell to other nerve cell. In this picture down here, you see an axon of one nerve cell, and it's coming in contact with the dendrites of the next nerve cell. So it still has to pass that information from the end of one cell to the beginning of a new cell, and then it has that gap in there. So we still have to go from electrical transmission to chemical to cross that gap, and then back to electrical again. So what we have to do then is we have to start thinking about this. We're going to order these things. So where is my synapse? My synapse is this gap, right, between two cells. So this would be one cell. This is the next cell that's receiving the information. So this one's sending info, and this one's receiving right here, okay? And so I have to get an electrical transmission from this one across that little gap, here's the synapse, to this one. And then that one would propagate a transmission along its cell. So in order to kind of effectively speak about these, I don't just say the first and the second. What we really say is the one in regards to the synapse. So here's my synapse, that little space. The one that's before the synapse is the presynaptic neuron. The one that's receiving or after the synapse is our postsynaptic neuron. And what is a presynaptic neuron in one interaction might actually be the postsynaptic in the next. So this, like this neuron right here. Let me erase some of this. So right here, I've got information coming from this neuron to this one. So this axon right here belongs to the presynaptic neuron. This cell body is the postsynaptic, but when it sends its information down its axon, we're going to end and terminate, and we'll have another cell. And so now it has become the presynaptic, and this is now the postsynaptic. So it's really just a relationship between that gap and which neuron we're talking about. Okay, is it before the gap, the synapse, or is it after the gap? All right, and then we got to talk about how that information is passed from one area to the next. So going back to those terms from like the first day of this class, I talked about IPSI and contra and all those kinds of things in that particular session. So an IPSI lateral response basically is when you have nerve cells sending information. So here's coming in sensory right here. Also remember that's afferent. We talked about in the last PowerPoint. So the sensory or afferent sending in information. It's being received right here. I've got an inner neuron in between and stuff like that, but I'm going to have this information go to a next. So the blue to the red. The gap is the synapse. This makes the blue sensory neuron our presynaptic neuron, right? And the red motor here is our postsynaptic neuron because it's after that synapse. But since this information is coming in from one side of the body and it's going right back out to the same side of the body, so like you stepped on a tack, your foot sends in that pain information, and then your foot you send out this information to raise your foot away from the tack. So that foot responds, pain from that foot, and then a response in the same foot coming the same side of my spinal cord. We call that IPSI lateral, okay? So same side of the body are being receiving information and sending information, but the synapse is sending it from the same side to the same side, okay? Contralateral means across. So contralateral, I receive this information here, and then it crosses over. There's your interneuron in between here. So I have a synapse between the sensory neuron, gap right there, and then the interneuron picks up the information, and then I have another synapse between the interneuron and the motor neuron. So here, presynaptic, and the little black one here, the interneuron is postsynaptic, and then between the little black interneuron and the red motor neuron, I have another synapse. So now the interneuron is the presynaptic, and the motor neuron is the postsynaptic. So get used to those terms. But directional. It's coming in from one side of the body, and then it's making an action on the opposite side of the body. So going back to that, like that tack, you picked up, or you stepped your foot on a tack. Here comes the pain sensation, right? From there, I will pick up that foot. That would be ipsilateral, but at the same time, the other foot, the other leg, has to do an opposite action than it was doing. Like when you're walking, right? You've got a step forward, and then you have the leg behind you, right? And then you change position so that those muscles relax, and you get a different set of actions. So those muscles go forward, right? Propelling your leg forward. Well, if I step on a tack and this foot now wants to pull up, I can't have this other foot continuing in a forward motion, because then I'd be on the floor. There'd be two feet in the air at the same time, right? So instead of continuing with that forward motion, now I have to put a direct motion straight back down on that leg. I've got to cause an opposite action on that leg than was previously occurring. That's this other leg now straightening out instead of bending to move forward. So that would be that contralateral. So I've got pain coming in one foot that raises that foot, right? And then an action that says put all my weight on that opposite leg to support the weight of that body as it's coming back down on it. All right, remember our neurotransmitters? We talked about those before, and remember I brought up that the main one in muscle contraction is acetylcholine. We're going to actually talk about acetylcholine again. So it is that acetylcholine. Don't forget that one. That's still a very important one. But now we're going to end up adding some more. There's a ton of these different types. There's a few that you need to know. Okay, acetylcholine is always one of those. Remember, here's my axon. So here comes my nerve impulse. Remember, when it gets down, the electrical impulse gets down to the axon terminal, the calcium channels open, calcium flows in, and the neurotransmitters then, there's that calcium coming in, neurotransmitters and the vesicles move to the surface and dump those little neurotransmitters in there into the synapse. They cross, they bind to little proteins on the opposite cell membrane. That would be the postsynaptic membrane right there. And then they're going to respond to those neurotransmitters. So still the same thing that we talked about with muscle, action from the nervous cell. There's still neurotransmitters, and it doesn't have to be nerve cell to muscle cell. It can be nerve cell to nerve cell or any other cell on the other side, as long as it responds to those neurotransmitters. Okay, I'm going to post this in D2L for you so that you can see this. And again, it's a nice animation. I would spend some time watching it because it actually shows the nerve impulse coming down. It shows the opening of these calcium channels. So the calcium flows in, then the neurotransmitters are moved down through the vesicles to the surface. They enter here, and then they bind to the postsynaptic membrane, the proteins on the postsynaptic membrane to cause that action. So really what's happening here, this chemical synapse, is change of electrical stimulus to a chemical stimulus, and then back to electrical again when you get to that other opposite end. This is presynaptic. This is postsynaptic. Don't forget those terms. This here, where the chemicals are acting, that's your synapse. But again, go through the video. All right, so we also have to just determine what's going on with our neurons and stuff. So we have a neuron. I'm going to draw you a very bad neuron right now. So here's my neuron. Okay. You got the cell body right here with this little nucleus. We've got dendrites right here. So here's the dendrites. They're going to receive information from another nerve cell or from an outside stimulus. They're going to transmit that information down through the axon, and then eventually it'll be picked up by another neuron or some other thing there. How does that electrical response make it down and travel through this neuron, this nerve cell, before it gets to that synapse? We talked about what happens at the synapse, but what happens right here? How does that information get from here to here? And what we have to do really is to create an environment around that nerve. It gets worse every time I draw it. Around that cell with ions. And what we want to do is make the inside of the cell have a little more negative charge, and the outside of the cell, the environment surrounding it, to have this more positive charge to it. That's electrical energy. Anytime I have a difference in charge, so if I have a difference in charge, okay, that's electrical energy. Whether it's powering your TV, powering your cell phone, whatever it is, powering your nerves, it's an electrical charge. And so this is how we're going to get that impulse to travel down through this cell, okay? Inside, more negative, outside, more positive. Guys, it's not 100%. It's not all or none. It's not all positive ions on one side and all negative on the inside. Just means that the outside has more of a positive charge and the inside has more of a negative charge compared to the surrounding environment. All right. Terms that you're going to need to know. When we have this set up, and this is positive out here and this is negative here, this is what we call a polarized membrane. Okay, so get to know these terms. I'm going to tell you there's polarized, depolarized, repolarized, and hyperpolarized. So you need to start getting the terms down. Polarized is positive outside the cell, negative inside the cell. And it has to do with charged ions. Learn that in chapter 2. So all of this stuff really does come back over and over again to you. I didn't teach it to you for no reason. See? Okay. So what ions are we using? There's a whole bunch of ions you could use because ions by definition have a charge. But your nerves use 2 specific ions for this. And calcium's another one that's going to come back, but not for this particular impulse, sending an impulse down a nerve. We've got potassium. So this K stands for potassium, by the way. All right. So the K is potassium. It has a positive charge. And the other one, the Na, is sodium. So we're going to use sodium ions. They also have a positive charge. So how is it I create an environment? Gosh, yeah, it does get worse and worse, doesn't it? An environment that's negative on the inside and positive on the outside if the only 2 ions I'm using are positive. It's not their charges that matter, it's their quantity that matters. So I'm going to put a ton of these positive ions on the outside and just fewer of them on the inside. That creates a difference in charge. I have a much more positive effect on the outside, a less positive effect on the inside, okay? So even though they're both positive, it's so that you can have, you just have less of a charge on one side or the other. And that does create this positive, negative sort of interaction between the two. This is going to require energy from the cell to do this because we're really trying to, pump out as much of these positive ions outside the cell as possible. And anytime I use a pump, that means ATP energy. Here's where those mitochondria come in. And so the nerve cells use a lot of glucose, use a lot of oxygen, use a lot of the mitochondria to do their work because they're constantly having to do a lot of work, especially with the sodium-potassium pump. So it's pumping ions out, and it's also pumping some on the inside too. So it's just moving them against their concentration gradient. Active transport, if you remember that from earlier in the semester. What we really want to do is we want to make it specific. I'm not going to just put a bunch of positive on the outside and a little positive on the inside. I'm actually going to stack the deck with specific ones. So what I want here is sodium on the outside and potassium on the inside. So I'm going to put a whole bunch of sodium out here as much as I can. There's that active transport, get it, just keep pumping them out of the cell. So there's a whole bunch on the outside and a few of your positive potassium on the inside, but I got to pump those in. So more potassium on the inside than you have out here, more sodium on the outside than you have on the inside. Both positive, but it still have a lot more positive surrounding the cell than what's inside the cell. Okay, so this, let's go back for 1 sec. I'm going to take a section of this cell that I drew. So from like here to here, we're going to cut that out, and that's what I'm going to use in the next slide. So when you see that, like the membrane here and the membrane here, you're looking at just this one section. So we're not looking at the whole cell. We're just going to take that one cell. All right. Remember, as we do this, that a cell is not 2 dimensions. It's 3 dimensionals, and it's really this axon would be like a cylinder. So think about a straw, right? So as you have the straw, we're looking at one side and we're looking at the other side of this, or this side and this side really doesn't matter. We're just not paying attention to everything else that's going down that tube. So I'm just kind of made it simplified and said, here's the 2 cell membranes, but they're really in 3 dimensions coming all the way around. Okay. All right. So here, this is one side of the cell right here on this side, and this is the other side. So this is the interior of the cell, and this is the exterior. So this is outside the cell, right? So what we want to do then is we want to get this cell polarized. And polarized just means technically it's at rest, but really it's not really at rest. What it is the neuron is primed. It's ready to send an impulse, but nothing's happening yet. So we have set the deck so that it's ready to do something, but it hasn't been told to do it yet. But you got to get it ready to do it, because if it's not ready, then nothing will ever happen. So what we've done is, this is where we put a bunch of these sodium ions outside of the cell, right? And then we've pumped some potassium on the inside, but not nearly as much potassium as we do sodium, which then makes this more negative, on the inside, and more positive on the outside. It's relative. Again, this is not entirely negative and this is not entirely positive. They're just in relationship to each other. This one has less of a positive charge on the inside of the cell and more of a positive charge on the outside. This is when my membrane is polarized. It is ready, it is set, but it is not go yet, okay? It's just ready to take that final step to say, okay, let's go and let's send that impulse. Okay, so like I said, more negative on the inside. Remember, that's because I put the potassium inside, which has a positive charge, but it's less of it. And a whole bunch of sodium on the outside, a lot more of that, more positive on the outside. It's charged, it's polarized, it's ready to receive an impulse from somewhere else that says, okay, it's my turn to do some work. Okay, and what does that look like? Okay, so when it's ready to do some work, I'm going to go back for a sec here. Okay, we have all along these membranes, remember in 3D, so it's going to be, like all the way around. We have like all of these little proteins. So you got little protein channels all around. Okay, I'm going to draw them on 2 sides. So I have protein channels here. I have one here. I have them all the way down, right? And I'm going to have them on both sides, but we're just going to look at it on one side. So here's my little protein channel. And a channel just means that it's like a tube that allows stuff to move through. My protein channels that are specific for sodium, so we're going to call them sodium channels, are closed, okay? That's keeping the sodium on the outside. If these channels are closed, sodium isn't allowed to flow through. But if these channels are open, sodium will freely flow through it. Likewise, I have little potassium channels that are here that are closed, and they're forcing that potassium to stay on the inside. What's going to happen is something happens to this membrane. You touch something, you feel heat, an electrical impulse comes in from another neuron. Some sort of stimulus occurs. And if it's enough, what it's going to do is open the first channel, or the first few channels. When that opens, sodium will start flowing in. And if enough sodium flows in, that cell then realizes it doesn't realize it's not got a brain, but it gets the information, the chemical information that says, I have enough of a change in charge from one side to the other, because now I'm bringing all this positive in here, that we're going to send this impulse. Okay, so potassium, that's the little black arrows right here. I'm sorry, not potassium, sodium, those little arrows right here, starts to flow from where it's here. It's going to flow in. So now the sodium is running inside the cell. Okay, in this area, I'm set at that charge where I'm supposed to be, at rest, polarized. Sodium outside, potassium inside. But now, when this first gate opens down here and sodium flows in, now I have potassium here plus sodium. So now I'm adding a whole bunch of positive. So now the inside becomes more positive because we're adding to the few potassium ions I had. I'm throwing a bunch of sodium in here. There's nothing out here. So this is now negative. And what I've done is I've taken my charges and I flipped them. So where I had sodium out here positive, right? Potassium on the inside, making that more negative in relation. Now I've taken that positive negative and I've just flipped that charge. Okay, that is an impulse. That change in electronegativity from one side of the membrane to the other is the beginning of an impulse. That's all an impulse is an electrical change. Okay, those ion gates open up, specifically the sodium ones, right? That allows the sodium to flow in, bringing more positive to the inside, flipping my charge. Okay. However, I've got to change the name. Remember, this is all polarized down here because it was positive on the outside and negative on the inside. That's what polarized means. Positive outside, negative inside, relatively. Now I'm negative outside, positive. I'm no longer polarized. I am depolarized because I flipped those charges. So all of this down here is depolarized. Okay. So that flip in charges has depolarized my membrane. This side's still polarized down here. It's only happening at this end because this is where my channels are opening, right? So here, and it's kind of traveling in that direction. So sodium flows in here. If enough sodium flows in here, it causes the next channel to open and the next channel to open, and we will eventually move all the way down that membrane. But right now, at this point in this picture, we're just right here. So polarized still here. So these guys on this end, they're not doing anything yet. They're still at rest. They're still waiting to be told to do something chemically. This area right in here, that area is the only area where my sodium channels have opened, sodium has flowed in, and I now have depolarized that area of the membrane. If enough of that happens, it will stimulate the next channel to open, the next channel to open, the next channel, and it's like dominoes going down. If you can think of, if you ever played dominoes as a kid, you have to set them up like in a row, right? That's, of course, a terrible picture. But you set them up, you expend energy to do that. That's ATP, right? So ATP to set them up. They're ready to go. They're at rest. They're polarized, but nothing's happened. The first thing that has to happen is you have to tip that first domino. And I can push that first domino and not get anything to happen. I have to push it enough that it actually pushes on the next one enough to get it to tip, and then the next one tips the next one and the next one. That's what's happening with our protein channels along that membrane. This is one protein channel, one sodium channel. Once it opens and enough sodium flows in, it tells the next one to open. The sodium flows in, it tells the next one, and you just keep flowing down that line, and that is a change in charge all the way down the line, sending an impulse. So here's terms also. It's sending an impulse, what we call a nerve impulse. It is also something we call an action potential. Okay, an action potential is the same thing as an impulse. When the membrane is polarized, remember it's at rest, we call that resting potential. Okay, so charged Sodium outside. I'm going to say this over and over again, guys, because you can't say it enough with it for y'all to get the idea. Sodium on the outside, potassium on the inside. We're at resting potential. My membrane is polarized. All of that goes together. Once the first sodium channel opens, sodium flows in. I have now depolarized my membrane, and I have begun to send an action potential, which again is that impulse. Lots of terms, fun. Practice them. Go through this, draw it out like I have it drawn out. Not that mine makes any sense anymore. All right, so again, first one open, made the next one open, made the next one open. All of this is sodium coming in, right? Keeps going on down the line, all the way in, all the way in, all the way in. All right, here's my problem. If I continue just opening channels and just having sodium flow in, and that's what you see right here, right? Our impulse will actually stall out, all right? So we have to force that impulse to continue all the way down the axon until it gets to the end. Otherwise, if I just open these channels and I've got all of this positive, 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 you know, unlike this picture, all the way down, it kind of just stalls out and it doesn't go any further. So what I'm going to have to do is push it from behind to keep that impulse going all the way down this axon, right? And the way you do that is you immediately behind. So this is where my sodium channels are open right here. This is my action potential occurring. Immediately behind it, I'm going to slam shut those sodium channels and open the potassium channels. So now my potassium will flow back out. And if my potassium flows out, now I've got positive on the outside again, and I've got negative on the inside. I have taken that charge from the beginning here. Here I'm at resting potential, and I flipped the charge. That was the action potential. And now I flipped the charge again and restored it back to positive, and I flipped it back again. The only place that you actually have an impulse is in between these two here. So it really started here, and it just kept traveling down, being pushed behind by this reversing of the charge, traveling down from step by step by step. So now you see it in this spot, and it's going to keep moving this direction. Okay, it's kind of like a wave. Think about a wave of water coming in. That wave moves across the ocean, right? It doesn't just like build up, build up, build up, and you have one. You have this wave that moves across as that water comes in contact with the water next to it. And this is what's happening here. All right. So here, oh, goodness, here I have potassium on the inside, sodium on the outside. I open my channels. So now I have sodium on the inside and potassium on the inside during my action potential. And then here on this side, now I'm throwing the potassium back out. So I have potassium on the outside, sodium on the inside. Have I restored back to where I started from, which was right here? That's a big no. Okay. Yes, I'm more positive on the outside. That looks the same right here and right here, and negative on the inside, but our ions are in the wrong place. So if I want to get back to the beginning again, back to my start again, I'm going to have to repolarize my membrane. So polarized, depolarized, and I'm going to have to repolarize my membrane, and that's by changing those charges again. And originally, and then finally at the end back here, if I really want to get my membranes all back to normal again, I'm going to have to use active transport to actually reverse this again. So I have to put my potassium back in here and my sodium back in here. The charges still look the same. See how the charges here, you know, when you draw it this way until you actually measure it, they look the same, but my ions are in different places. Okay, so now I'm back at resting potential once that's all been restored. But this is just a repolarization. This portion right here is a quick fix to force that impulse to keep going forward. If I don't do that, if I don't force, repolarize, if I don't do this opposite, throw this potassium back out, that's when my impulse will just stop in its place. So this is just to force it. This is a quick fix. I get back to resting potential again. That's just restoring it back to normal, ready for the next impulse to come through. It's really confusing. It's really not a lot of things going on. You're just moving potassium and sodium back and forth, but it's confusing as to the order of these things as they're going along in here. All right, so here's the sodium potassium pump. That's going to use ATP. This is active transport. This is only when I am restoring to resting potential, okay? So this is really after an impulse has already gone through, it's passed, and I want to get to the point where I'm ready to be at resting potential so I can receive another impulse that will send another action potential, okay? So the sodium potassium pump uses ATP to pump 3 sodium outside the cell and two potassium inside the cell. Again, positive inside, positive outside, but look at the numbers. I've got 3 sodium to only 2 potassium. That means there's less positive on the inside, more positive on the outside, giving this a greater positive charge and that a greater negative charge in relationship to the outside of the cell. Okay, and these are just showing some proteins. All right. All right, so let's talk numbers then. So the next thing is going to be numbers here. Remember our resting potential, our resting membrane potential, but it's shorter to say resting potential. So our resting potential has to have, here's my cell, positive on the outside, negative on the inside, right? The difference in charge from out here to inside is a difference of 70 millivolts. So what we say when we talk about this little cell right here, I need a little bit bigger space to write. So here's my negative, is it is actually negative 70 millivolts of charge compared to the outside. So you're going to say, see where it says 70 millivolt difference? There's a difference in charge from outside to inside, but we will always refer to the inside of the cell compared to that. So our resting membrane potential is actually negative 70 millivolts because it's 70 millivolts less than what's on the outside, okay? And that has to do with this distribution of these sodium ions on the outside, potassium ions on the inside, more positive out than you have in, okay? And all of this is set up by that sodium potassium pump, again, ATP, active transport. Also remember that's going to be the mitochondria doing this kind of stuff, providing that ATP for that, okay? All right, and here you can just, I've got a picture here, better than my pictures, negative on the inside, so this is negative 70 millivolts compared to the outside. So it's difference of charge of positive to negative, it's just 70 millivolt difference. Resting potential, ready, set, but not go yet, right? This is waiting to receive information to say, let's send an impulse. All right, so what can change those charges? What does it mean to receive information to say, start opening those channels so that sodium will start flowing in? It's any stimulus. It can be a difference in temperature. So you touch something hot, you touch something cold, whatever it is. Temperature, you've got receptors that pick up a temperature change and say, it's time to react to this. I've got to understand that this is a hot substance or a warm substance or a cold substance, and my brain has to figure that out. So a temperature change is going to stimulate certain receptors to start making those changes and opening those sodium channels. It could be a different set of receptors that respond to light, your eyes. So your eyes are going to have receptors that light will say, now it's time to open those channels and send that sodium in. And likewise, pressure is another one. So if I put my hand down pretty firmly on the desk or somewhere, I'm going to stimulate some pressure sensors. That's their only job, pressure. And then that will actually change the shape of this protein channel, and that causes it to open, and then sodium can flow in. Environmental changes can also affect it. So the gate that opens here. So I can have like this first one, maybe it's a pressure sensor in your fingertip, right? It's closed, and I put enough pressure down on it that it opens this channel up, and then sodium can flow in, right? But that's only the first one. How do I get that impulse all the way down that neuron? The next channel in line isn't necessarily going to respond to pressure. It's got to respond to a difference in charge from this one. So it's relying on information from this channel right here. So as that sodium flows in, it's going to say, okay, that one responded to this change. Now I need to respond to this. And then we have that domino effect of everything else working with that. So they can be chemically gated. That would be at the end of, here's a neuron, right? The synapse right here. So as the neurotransmitters come through, they open in response to neurotransmitters, like acetylcholine would be one of those, that cause those gates to open. That's a chemically gated channel. A pressure gated channel won't respond to acetylcholine. They have two different effects, basically. It can be voltage gated. Remember this one here that I drew in over here, respond to pressure. But the next one in line is going to respond to a difference in charge. Now that all that sodium's flowing in, it says, oh, there's a difference in charge. I need to open up and more sodium can flow in. So that would be a voltage gated channel. It's responding to that change from that -70 to something else. Or it can be mechanically gated. Mechanically gated is like this, like pressure right here. If I put pressure on it, I've physically changed the shape of that channel, and now it opens up. Okay. This is showing kind of a sequential 1, 2, 3 sequential impulse here. So down here in the blue, this is where I would have a resting potential. My membrane is polarized. Remember to start using your terms. And what that really means is I have sodium out here and potassium inside, right? In the pink, these little symbols stand for electrical charge, dipoles and stuff. In the pink, what's happened is the sodium channel opened, sodium flows in, so now I have sodium and potassium on the inside because the only thing allowed to move is sodium. This, remember, is called our action potential or impulse, right? And what I've done is I have depolarized my membrane in that pink section. And then behind it, so that I can keep this moving this direction here, behind it, my potassium channel's open and potassium flows out. So now I have sodium in here. So I've got sodium and potassium right in this spot, right? I've got sodium over here and potassium right here. So this is still your resting potential, right? This is your action potential in pink. And then behind it, I still have sodium here, but the potassium flows out. So now I am repolarizing my membrane, resetting those charges. So now it's positive on the outside, negative on the inside. But I really haven't gone back to normal yet. I've done a quick fix in order to make that impulse, push it from behind to keep it going in the direction I want it to go, instead of just kind of stalling out and not going anywhere. If it stopped right here, it's a dead end. Can't do anything. And then that impulse will keep going, and these channels will then, these channels are opening here for sodium. So sodium's coming in. The sodium channels are closing here, and the potassium channels are opening. So you've got an opposite thing going on. This is sodium open, right? And potassium closed during the action potential. In the back side of this is potassium open and sodium closed, right? And then right back here at the beginning, to restart it, I'm going to do that sodium potassium pump, use ATP, and that will restore everything back to its beginning again and put the sodium on the outside and the potassium on the inside. So 4 really steps going on in there, but that all keeps that unidirectional flow of that impulse. All right. So we've had polarized. That's that resting potential. Sodium outside, potassium inside. We've had depolarized. That's an action potential, and that's sodium inside and potassium inside. And then we had repolarized. which is restoring, so the charge, basically flipping the charges back. So that's potassium on the outside and sodium on the inside. And now we've got a new one, hyperpolarized. Hyper means above and a lot, right? So hyperpolarized means, remember I had this difference in charge where it was negative 70 millivolts of charge difference between the outside and the inside? Hyperpolarized means I went way above that. So it's not negative 70. I've got it at something like negative 90 millivolts. Too much of a change. I want this. Hyperpolarized is going a little bit above that. So more negative than it's supposed to be. All right. And this is where everybody gets confused because you think in positives, but this is more negative than you want it to be. Think about your bank account. A lot more, negative $10 versus negative $200. That's going to be a difference to you in your bank account. But negative 200 would be the hyperpolarized there. All right. So all of these terms need to be something that you need to be practicing as you're going through this. Okay. Introduce this term in the last chapter too, threshold potential. I can actually open some of these channels and get some sodium to flow in, but I really need to have enough flow in to actually stimulate the next channel to open and so on. So I can get some change in charge without really providing for an impulse to occur. That's threshold. Once I reach that point where the next one is actually encouraged to open, that's threshold. Okay, so it's that all or none thing where you can give a little bit of change, but it's not enough change to get it started. And I've got some graphs on the next page. I'll show you that. But if I reach that point, it's all or none. I can't stop it at that point. If I get to threshold, my action potential is going to be propagated and it's going to be sent down that neuron and there's no going back. Okay, it's going to send that action potential. Again, those are just potential changes. So at rest, we're going to have a negative 70 on the millivolts on the inside compared to the outside. As I start changing charges, that number is going to change because now I'm going to make it more, I went from positive on the outside to negative. I'm going to flip that. So it's not negative 70 anymore. That number is going to change. All right. Okay. So at rest, or resting potential, your membrane is polarized. And that's a difference in charge from the outside to the inside of negative 70 millivolts. So here on this graph, you can see this right here. This is a line showing at rest, negative 70 millivolts. This is negative 70 on the inside. All right. We're ready to go, primed, nothing's happening. An impulse comes in here. Some sort of stimulus comes in here and starts forcing those gates to open up. That means that I am going to start putting sodium inside, change that charge. Remember where I had positive here, negative here. Now I've got negative here and positive here. Change the charge. You can see it in the graph. So here's it sitting there at resting. And then I start changing my charge. This all yellow right here is sodium flowing in. Notice it goes, this is a 0, it says negative 0. I don't know who made that graph. I got it from the textbook or I got it from Google, I don't know. But anyway, it goes to 0. There is no negative 0. It goes to 0 and notice that it goes above that. So we're really changing that charge. We're not going to even charges on both sides. That would be negative. That would be equal, right? What we're doing is we're making it more positive on the inside. So we have to go to a charge that goes above 0, rises above 0. There's this point right in here at about negative 55 millivolts that is threshold. If I have a change in charge, so some sodium starts to flow in, but not a lot flows in. If I never reached a difference in charge of negative 55, my impulse just stalls out. It's done. It's not going anywhere. But once I reach negative 55, everything starts happening. All those channels all the way down the line are going to start opening up and there's nothing you can do. That's my threshold. So that negative 55 is the do or die point. If it's below and never reaches to negative 55, no impulse. Once it reaches 55, all those little channels all in order start to go, okay? And everything starts to depolarize. We move towards 0. We move past 0 towards a positive, okay? So here's that sodium flowing in, right? Sodium flows in first, and then potassium will flow out. So looking at my graph again right here, resting potential down here at negative 70. Sodium is flowing into the cell as this line is rising up, and it goes up to a positive interior charge of 30 millivolts compared to the outside, okay, with all that sodium in there. So at this point, I have sodium and potassium, by the way. So sodium and potassium are both in here, creating that extra positive charge. Once it reaches 30 millivolts, that's when the sodium channels close. So sodium close, potassium open, and the potassium flows out. And once the potassium flows out, notice that the reverse in charge happens again, and we go back down to negative. So we quickly flow back to a positive outside, negative on the inside. So for, we started positive with positive outside, negative inside. Impulse, we reversed it, negative outside, positive inside, and then we reversed it again here. So this portion right here where the sodium is flowing out, that's depolarization. This portion right here where the potassium channels have opened and the sodium have closed, this is repolarization. And notice that when it gets down here, it actually drops below that negative 70 and it gets to about negative 90 millivolts. It hyperpolarizes for a moment. Look how quick that is. It can't stay that way, so it just does it like overshoots the target and then comes back again. And it's going to reset to negative 70 millivolts and it's ready for the next impulse. We got to set back to resting potential. Why? Why can't it just stay at negative 70? Think about threshold, right? Think about what would happen if it stayed down here at negative 70. Negative 55 is my threshold. I have to reach that if I really want to send that impulse. Otherwise, that impulse just dies out. Well, if I start all the way down here at negative 90, I've got that much more territory that I have to cover to even get to threshold. So you're unlikely to actually meet threshold in order to send an impulse. So if you stayed at too low here, too hyperpolarized all the time, you would have less impulses sent, less stimulation of muscles. So that's a problem, and that does occur. And that is a problem with sending that impulse. So we really want things to be at the right amount. You want it to be at negative 70 at resting potential. You want it to hit negative 55 if you send that impulse. You want it to go to 30, and then you want it reset back at negative 70 again to hit resting potential again. The hyperpolarization for a moment's okay. Overshoot, but get back stabilized. Here's a bigger picture of that. So once again, I'm going to walk you through it. Here's my negative 70. This is resting potential. This is when my membrane is polarized. And what I have is sodium out, right, and potassium inside the cell. Stimulus comes along, sodium channels open, sodium starts to flow in. If it never gets enough sodium in, the whole impulse will just die out. It has to reach threshold at negative 55. So when it gets to negative 55, boom, there's no stopping it. And it quickly, all of the other sodium channels open up. We get up past there to 30 millivolts. So we're in the positives. We've reversed our charges. This was an action potential. Remember that term. And I have depolarized my membrane. It hits 30. Quickly, my sodium channels close. My potassium channels open. Potassium then leaves the cell and goes outside when we reach that 30. And boom, it starts to repolarize very quickly, okay? Hyperpolarizes to negative 90, but then it resets itself, and we reestablish resting potential with the sodium-potassium pump, ATP, right? And now we're reset at sodium out and potassium inside the cell. And now it's ready back at resting potential ready to go again. Okay, it does take a little while. Notice that. Notice how quick the impulse sends, and then it takes a little while to reset. So it does have that issue going on there, but that's okay. Works for us. And this is just showing the same thing. Here's my charges, resting potential, as I've got sodium out here, and I've got potassium in here. We got some sort of stimulus that's opened up a channel, right? Sodium flows in. This is the beginning of my action potential. This is depolarization, whereas this was polarization, right? Action potential moves down. So one channel stimulates the next channel, stimulates the next, stimulates the next, and that's what's happening here. And from behind it, now my potassium is flowing out so that I can repolarize and shove that impulse down the line. Keep it moving down the line. Repolarize. So polarized, depolarized, repolarized, and then reset with the sodium potassium pump at the end. And then you're ready to go for another impulse. And that's it for today. I'll see you on the next video.